from monkeys and apes into humans at least a dozen times in history, but actually quite recently. So different species of monkeys have infected humans because they were hunted and butchered, as well as some subspecies of chimpanzees. Now, almost all of these species that are naturally infected either don't get infected, or if they do get infected, they don't get sick. They have been evolving and co-evolving with these viruses for hundreds of thousands of years to literally millions of years. So they are basically immune to this. And so we, in our lab, have been not only working up their evolutionary history and estimating the virus's evolutionary history and see where they cross paths, but by looking at specific proteins and receptor molecules, different components of the immune system, we are looking to see, are there specific changes in different lineages of old world monkeys and apes that seem to confer them immunity to the AIDS virus. But the only way to figure that out is by knowing their evolutionary history and the virus's history and figuring out which additional changes aren't explained just by their normal evolutionary history. And we've been making great progress on finding certain genes that seem to confer resistance or different components of the immune system that are more or less important. We're hoping vaccine researchers and other biomedical researchers will be able to use some of this information. You may have recently, just literally in the last several days, heard stories that chimpanzees at Gandhi, the famous site where Jane Goodall works, actually turn out they do indeed get sick and die from an AIDS-like condition. Well, it turns out, so far, it only looks like one specific subspecies of chimpanzee does this, not all of them, and that this subspecies, which was a little bit more isolated, has actually only been infected for several tens of thousands, if not hundreds of thousands of years, as opposed to millions of years like their immune brethren. So we actually have an unfortunate test or an example of what it is like for a species to be relatively newly infected. So we can look at the differences in this subspecies of chimps versus the subspecies that are immune and maybe even get a quicker and stronger signal of the specific changes that lead to immunity of this virus. We are also working on malaria in my lab. Just like AIDS, there are different species and subspecies that are very closely related of monkeys and apes, that some of the subspecies get very ill and even die from malaria, and others don't get sick at all, or they have a very mild condition. So by comparing the species that get sick with those that don't, looking at their evolutionary history, and again, looking at receptor molecules and genes in their immune system, we might be able to find some answers in how to deal with malaria, which is, again, one of the really nasty, horrible diseases. So part of the message here is, even though I'm an anthropologist, we're actually doing the latest in bioinformatics, the latest in molecular biology, and we are assisting with biomedical research. So whatever field you might ultimately go into, don't track yourself too narrowly. You can find that a field that at first doesn't seem like it's going to be relevant, but you're really interested, might turn out to be relevant. I never thought when I started anthropology, and I actually started out looking for fossils, that I would be doing AIDS and malaria research. But that's the joy of research, is that you can branch out into all of these different fields and subfields. We also do something called conservation genetics. By determining the variation found in different populations and subspecies and new groups of primates, we can find out if, by creating a national park around them, that they have a good chance of survival, or unfortunately, sometimes we find out they don't have a good chance of survival because they just don't have enough genetic variation left in them. Or when we find a new population, we can determine if it's indeed truly unique and separate from other ones, and therefore perhaps more worthy of our efforts, or if it's just another population of one that's already relatively healthy. Because the unfortunate fact is we have limited resources to apply in trying to save these endangered species, and we want to put those resources where they are most efficient. We're doing a neat little study in the lab right now as well, in which we are genotyping, and actually it's a forensic study of Diane Fossey's gorillas. These gorillas were buried in graves at her research site in Rwanda. But through the last 2 decades of civil wars, much of the records have disappeared, the markers have disappeared. So we are going back and actually trying to identify individual animals for which there are very extensive behavioral records, so that we can look at different aspects of their skeletal growth and development to see the diseases they had, how that might have affected their teeth and their skeletons. But the first step of this project is actually to work on, to identify the individuals and go back to archived blood samples and hair samples and things that were brought to Europe in the 60s and 70s. And so it's sort of a real forensic case. It just happens to be on gorillas. But gorillas are so closely related to us, as are chimpanzees, that we are using all of the DNA markers and reagents we're using are the very same ones they use down the street at the medical examiner's office. We haven't had to shift our protocols at all. We're using the very same DNA fingerprinting techniques as you would use on humans, but in this case in gorillas. We also like to have fun in our lab, and Dr. Satt had alluded to the fact that we've done a lot of different TV shows. So using the very same techniques to identify a new species of primate or to figure out relationships, these sort of forensic DNA techniques, we have been testing monsters. So we have done lots of Bigfoot, Yeti, Sasquatch, the Chupacabra. So far I've found bears, pigs, coyotes, people, synthetic hair, so I don't know if it was Barbie or Ken, but the chupacabras that we did turned out to be coyotes with mange. So in over a dozen different purported monsters, the orang pendek of Sumatra, the almas of southern Russia, in all of these cases, we found that they were either just merely humans or other animals, and we're continuously testing different things to see if, we have the find of the century or not. But I believe in doing these studies because we can sort of debunk poor and weak and weird science. You have people out there proposing crazy theories, and by throwing scientific methods at them, we can say, no, we don't have any evidence of a 7-foot-tall ape running around the Pacific Northwest, or the Jersey Devil just across the river, Montauk monster found earlier this summer. When we actually apply DNA tests to these things, we can say, look, this was fake. This was 2 different animals glued together, and that's why it looks so strange.